Hi everyone, my name is Renuka Kudva and I'm a postdoctoral researcher at Stockholm University in Sweden, and it is my pleasure to be able to talk about the work that I've done during my postdoc and the work that's generally done in my lab. So we're all familiar with proteins. They form an important part of our diet and we consume them in copious amounts because they're healthy. But proteins are also produced in our own cells and they fulfill several roles. They form structural elements that maintain cell shape. Enzymes that maintain cellular functions are proteins. Proteins power our muscles to keep us moving. They keep our blood flowing. Proteins keep us healthy, but they also make us ill. So our cells produce proteins all the time, and they're produced rapidly. These proteins are produced by stringing together amino acids, the building blocks of proteins, to form peptides that ultimately assemble into proteins into their 3D final state. This stringing together of amino acids is performed by ribosomes that are present in all our cells. They are universally conserved. And these ribosomes decode the nucleic acid mRNA that is encoded by the DNA, which is a molecule that we are all familiar with. So proteins produced in cells need to fold correctly to execute their function. The information on how proteins need to fold is encoded within the sequence of the protein itself, and this is called Anfinsen's principle. Now the misfolding of proteins can occur, and this contributes to several diseases such as ALS, Parkinson's, Alzheimer's, and even cancer. So it's very important to understand how proteins fold so that one can understand how proteins misfold and conduct research on the area of disease. Protein folding itself has been rather challenging to study because classically it involves isolating proteins from their native cellular environments and putting them into tubes, unfolding them in a buffer, and then observing how they refold in time. So this kind of sheds light on how proteins fold, the pathways that they take, that is, and how fast or slowly they fold. But time scales are rather different in our cells, where processes need to occur rapidly so that cells can be kept alive. And the cellular environment is also quite different from the environment that the proteins encounter in the tube. Cells are first of all more crowded because they are protein rich and there are lots of ribosomes. And what would make the most amount of sense is for proteins to be able to fold as soon as they are synthesized in order for them to be able to fulfill their function as soon as possible. Now this process would be termed cotranslational because it would occur coupled to the translation of proteins. And this field has gained a lot of attention recently because it potentially reflects a physiological state in the cell. So we use the gut bacterium Escherichia coli as a model organism to study how proteins fold cotranslationally, because the beauty of this process is that it's evolutionally conserved. And what you can study in a unicellular organism like the bacterium also probably holds true in our cells, but in a slightly more complex environment. So E. coli is really rather simple to grow and cheap to grow, and we have been able to get a lot of information out of E. coli that can possibly be applied to our cells as well. So our research so far has demonstrated that a large number of protein domains that form larger complexes of proteins, can fold cotranslationally. And we've used a combination of biochemistry, where we've pipetted stuff together in tubes, and structural biology, where we've been able to visualize a ribosome that's translating these beads of amino acids to give rise to a protein at the exit that folds. So we've actually been able to catch this in the act. So our hope for the future is that we're able to actually apply this also to our cells, and to a large extent in the future be able to study how proteins fold versus how they can misfold, and thus shedding light on the process of disease. Thanks very much for listening.